Okay, good.
Thanks for the nice introduction. Good afternoon, everyone. It's my pleasure to be here to share my current work. So I'm looking forward to talking with you about machine learning guided direct evolution of halogenesis for biocatalysis. So in the pharmaceutical and the agrochemical field, there are lots of compounds harboring the halogen atom, because if the compounds introduce the halogen in it, it would have the profound effect on the biological activity or the bioavailability. And also such compounds is also a very valuable building block which could be involved in some chemical reactions, for example, the cross-coupling reactions. So also besides the chemical synthesis, also in the nature, we also could find some halogenated natural products, which with the potent bioactivity. So in the nature, so the nature offers us the halogenase to perform the halogenation reaction. So there are in total 4 types of halogenase. They are haloperoxidase, flavin-dependent halogenases, also the alpha ketone turret dependent halogenesis, and the sum dependent halogenesis. And in each class, depending their substrate scope, they also have like the subscope, the subclass. And except the enzyme could accept the small moleculars. If the enzyme faces some challenge to do the halogenation reaction, the enzyme would prefer that they use a carrier protein to denote the other difficult substrate to perform the halogenation reaction. For example, the substrate about proly, also the phenolic ones. So even the nature offers the Y type to us, but sometimes that Y type cannot fully satisfied for our application. Then the direct evolution is a powerful tool for tailoring the enzyme properties, then achieve our target. However, that's in the traditional direct evolution, we still face some challenge. For example, the time-consuming, also rare beneficial mutants could be obtained. So recently, the machine learning provide us a solution to overcome these drawbacks. In this project, our objective is developed an AI-driven, fully automated and closed DBTL circle for enzyme engineering. Of course, here we use the study case is the halogenesis as a study model. And we want to figure out the scientific question about the enzyme specificity and the selectivity. So after we set up the pipeline, then we want to integrate those machine learning combined with the automation. So here, here is the IBL5. And then we can isolate the development of enzyme engineering, also for biocatalysis part. Yeah, so here shows our workflow for when we start the specificity and selectivity from the halogenous substrate and the product database. So if we want to train a machine learning model, that's the first step is we get a qualified database. So regarding the source of data, we either can get from the current database or from the literatures, or we can generate them by ourselves, experimentally. Yeah. After we get those data, after data cleaning and unification, we can use it for machine learning training. So here, for the application courses, we want to figure out the halogenation specificity and also the selectivity problem. So in the end, our vision is, could the model also give us suggestions like which enzyme and which substrate we could use them for the halogenation reactions. Yeah, of course, here we should use experimental validation to validate our training model. And then those data could feed the source of data again. Then we can get a really closed loop circle. So by searching for the literatures, we identified in total about 243 sequences, like they are all halogenesis. And we also label them, like link them with the UniProt ID. Then in the future, we can get enough features from the current database. And here we did one sequence similarity network analysis. So here, each dot means one specific sequence. So from the color, you could see the mixture clusters, which means the like different enzymes they are could cluster together, and then this mixture cluster holds the potential for the broad substrate scope. Yeah. So in the database, which information we want to get. So first of all, it's the protein sequence, and then it's the substrate information, also the cofactor, of course, also the product information. So here we use the smells information for the substrates, also cofactor and product. But except the current database and literature, we can get like very high quality data for our in-house experimental data. So limited by the high throughput screening method or the really high quality data, we currently only could know the information about the product, like it's yes or no. It's like if this substrate could do the reaction or not. But if you want to know which position we got the halogenation. So this is quite hard because currently, like we should use NMR to really identify the specific structure of the product. So which means, we also have some limitation if we really want to go the high throughput data site by ourselves, yeah. So when we get the data sets, then here I show the machine learning workflow that how could we use the data sets. So currently we are trying that towards the substrates, we use the message passing neural network for embedding the substrates. And in terms of the halogenesis part, we use evolutionary scale modeling and to embedding this halogenesis. After we get the average embedding, then we use them to fit the fully connected neural network, and then we train the model. So since we still like building up the data sets, then we just do some, we did some like preliminary test. So here we use the input is the reaction. So here we only use the information about substrate and also the enzyme sequence. And the output is whether this substrate could do the reaction or not. And this data out, So for the training data set, we get the data from the Brenda. Also, we extract some data like manually from the literature. And for the test data set, we did some in-house experimental, we got some in-house experimental results. So from here, we could see currently the model is, didn't work so well. For example, the training loss is not, it didn't like decrease significantly. Also, for the data validation, for the results of the validation data site, it's just like achieve the random algorithm. So which means we still need to work more on this part. I think this kind of like maybe limited by current data site. Also, maybe we can do more modification for the models. Yeah, this part is mainly supported by the Yufeng Su. Is one graduate students from Professor Peng's group. And yeah, so let's talk about the experimental validation. So to get such high throughput reactions, we should set up like a general experimental pipeline. So here we set up the general experimental pipeline for like a to get the purified enzyme. Yeah, so currently we could successfully get 38 well-expressed halogenesis, and they could be used for the in vitro reaction test. And we could see here they have very good diversity. They include all 4 types of halogenesis. Yeah, here the SDS page shows the purity of the enzyme of the protein is quite high. So when we get the purified enzyme, then next step is to perform the reactions. So if we really want to get high throughput, I think automation is a good solution. So here we set up a high throughput characterization on the ABAFAB, like base. So here it's mainly focused on the flexion dependent halogenesis. Yeah. So this part could be done on the TCAN fluent. Is this part. So after we like perform the reaction, then we can do the AC like after 24 hours after AC. So then we could use the LC mass to detect the results. Yeah, so here I would share a video like about the automation. So regarding the time limitation, I think we only could, I will only show you shortly. So here, I think the principle for this is it's really simulate the real researcher, how could we do the reactions. So for example, in this case, we get the substrates, we get the enzyme, and then we mix them together. So then I think, yeah, here is 3 speed, 3 times speed. So we could do the 96 reactions like in less than 10 minutes. But if we manually do it, I think at least maybe half hour for one researcher. Yes, okay, so let's summary. So until now that we already set up the pipeline are for the, for one part is the data extraction and also the ML workflow. And of course, for the experimental validation parts, we also set up the automatic pipeline. And for the outlook in the future, that we still need to enrich our halogeneous substrate database and get more data from the literature. Also, we're also thinking about like get also more features from the structure side. And then for the machine learning models, we could try more. And even like I got the idea from yesterday's talk, like Dr. Jiang, if I remember correctly, it's like how to perform the reactions more R. I think the work from the Professor Burke's group, yeah. And then so those work is mainly focused on the Y type, right? But as I mentioned, the Y type cannot satisfy for our application. And the next step is to do the engineering. Then currently we also like working on building up the automatic protein engineering pipeline, like to automatically generate the mutations and then perform the AC and test their properties. So in the end, I would acknowledge my advisor, Professor Huimin Zhao, and to bring me to this project, and also other professors like Professor Denmark, Burke, White, Professor White, and also Professor Shukla. And We have like a great discussion here in the monthly meeting and give me a lot of valuable suggestions. And I would also thank my co-worker Yufeng Su and Tian Hao, and also the co-workers in Dr. Zhou's group. Yeah. In the end, I would thank the funding part, the NSF, yeah, also the MMLI. Yeah.
Okay, in light of the time, I think we can try to fit in like one question before we go to the virtual poster session. It's also the last time to, win the question of the day award for the students and postdocs in the audience. So
you can only win one, by the way.
Yeah, that's good.
Take the chance.
I already won one, so I'm scooping someone's opportunity here. But so when you showed the work from the Peng group in collaboration,
yeah,
it didn't look like your neural net was converging at all, and it looked pretty similar to the random, I think, was what you were showing there.
Yeah.
And so, remind me, are you just feeding in the primary sequence of the protein to the neural nets, or how exactly do you featureize it going in? And do you have any sort of, I guess, do you see that as a problem with your data set, or do you see that as a problem with the representation of the protein?
Yeah, this is a great question. So actually for that part work, at the beginning, that pipeline, we use it for another enzyme is called P450. And that one, like in the database, we also include the information about the inhibitor. But when we just simply transfer those model or those pipeline to our heterogeneous ones, then the results looks not good enough. So I think the basic problem is the behind the logic core. So for example, for that one, or for example, for our case, we don't need the inhibitor. But for the P450, they need those data. So which means if you simply just transfer it to another project, so it looks not work well. Yeah.
Okay, so basically, you're trying to use it out of the box with a different optimization and
yeah.
Okay,
yeah.
All right, great. Thank you.
Thank you.
Yeah, thank you, Ocean, again.
Thank you.
And please join us on GatherTown for the virtual poster session, basically right after this. So, thanks, everybody.